Kia ora, my name is George and I've been spending the last few months working at the University of Canterbury looking at slime molds. So, slime molds are my favourite living things on this planet, and I'm going to try and convince you why they should be yours too. Before I show you what I've been up to with the slime molds, I'll give you a quick rundown on what a slime mold is and why you should care about them. Slime molds, or myxomycetes as scientists tend to call them, are giant amoeba that generally live in soil and on rotting logs and leaves. They're often grouped in with fungi, like moulds and mushrooms, due to how they look and where they live. But slime molds aren't fungi, and unlike fungi, they don't decompose things. Instead, they hunt. Tiny life forms called bacteria are the primary food source for slime molds. These bacteria are absolutely everywhere and they grow really quickly. A lot of them, in fact, make you sick. And to see a single bacterium, you generally need a microscope that can magnify something up to 1,000 times. And yet they're hunted by these slime molds, which, as these pictures show, are actually quite easy to find and quite easy to see. Slime molds can be millions of times larger than their prey, slowly creeping along the forest floor, consuming all the bacteria. Now, from my point of view, that's really cool, but if you were a tiny little bacteria, that would be truly terrifying. Now, most slime molds that you'll be able to find in nature are called plasmodial slime molds. These are the largest type of slime molds, and they have three main stages in their life cycle. They start life as tiny spores which will hatch into small single-celled amoeba that will chase bacteria around. These are really cool, and there's lots of other organisms that do this. But when slime molds are special is that they come together to create a larger thing. So amoeba will find each other of the same species and they'll fuse, and then it creates a larger amoeba, which is called the plasmodium. The plasmodium is still a single cell, even when it grows to the size of multiple square centimeters, making them some of the largest cells in the world. The plasmodium will keep feeding and growing until it either runs out of food or conditions become really unfavorable, such as it's too dry. And when this happened, the slime mold will form tiny mushroom-like structures called fruiting bodies, and these hold the spores. Generally, the wind will come along and disperse these spores. Different species will have different fruiting bodies, and there's quite a lot of variety, which is what we use to identify different species of slime molds. So for example, this is the dog vomit slime mold, which is easily found on mulch or pine needles, and it has one of the largest fruiting bodies. The chocolate tube slime molds, pictured here, have lots of thin brown stalks which turn into dust when you touch them. This helps the wind blow their spores to new environments where they can repeat their life cycle. Now, what if I told you that these strange microorganisms, which grow into large blobs of a single cell, seem to be intelligent? There's a famous experiment with the slime mold Physarum polycephalum that used it to recreate the subway system in Tokyo. This experiment showed that slime molds are great at optimizing. They'll always find the shortest route between two pieces of food. Other experiments have showed that slime molds can remember things, can learn things, and people are even trying to use them to build computers made out of living things. There are other types of slime molds called the cellular slime molds that are studied for their ability to work together. Dictyostelium discoideum, try saying that one quickly, is a cellular slime mold which doesn't make a single plasmodium. Instead, many small amoeba band together to make a big ball with lots of individuals, and this lets them move faster and find more food. But when the food runs out, some of the slime molds in the group will form a stalk, and others will form spores, much like the fruiting bodies of the plasmodial slime molds. The cells in the stalk die off, while those in the spores survive on. Outside of scientific research, slime molds are also used in classrooms overseas. This is because they are safe, fun, and easy ways to demonstrate a lot of biological concepts. Physarum polycephalum is used all over the world. However, this slime mold has never been found in New Zealand, and that means that we can't use it in classrooms or in scientific research without going through a whole load of paperwork. So that's what my project was about, finding a native slime mold in New Zealand that could be used for similar experiments. This started by going out and collecting slime molds. So I'm going to go through my process of collection and then how I actually managed to grow the slime molds and what I did with them. So I went out and looked for slime molds in forests and parks. The best places to look are on logs and on wet decaying leaves, as you sometimes might find a plasmodium. But most of the time you end up finding fruiting bodies, which can be really small or can be quite large. Once you've found a few, it's quite easy to find more. I found quite a number of fruiting bodies and a few live plasmodia, and I took these back to my lab and I put them on agar and gave them oats to eat. Oats are a favourite of Physarum polycephalum, so if I can find slime molds that also like oats, they should, in theory, be quite similar. Another way to collect slime molds that I use is called the moist chamber method. This is where you take a twig or some leaves, you put them in a container with wet paper towels on the bottom and you put a lid over it, and then you check it every few days for a couple of weeks. Sometimes slime molds are out there in nature, but they aren't actually growing into anything big. They're not forming big plasmodia or fruiting bodies. But if you put them in these conditions, eventually they'll all group up together and they might make a fruiting body that you can find. I found plenty of fruiting bodies and even a few live plasmodia. These were then taken back to the lab to be cultured. They were put on agar and given oats to eat. Oats are a favourite food of some slime molds, like Physarum polycephalum. From this, I managed to get 2 slime molds growing. These are both plasmodial slime molds and are easy to keep alive with oats and water. Once they've eaten all the food on their agar plate, I put them onto a new one so they can stay in their plasmodial stage, never running out of food. I can also split them in half to get more and more slime molds, which makes repeating experiments very easy. Here's some cool photos of the yellow slime mold, which is called Badhamia utricularis. I found this out by looking at its morphology, so how the slime mold looks, what features it has, and by analysing its genetic code. The white slime mold has been much harder to identify, but it's likely a member of the genus Physarum, along with Physarum polycephalum. So these are very closely related slime molds. They're all very similar, they look relatively similar, and they should do similar things. So I put it up to the test and set up some experiments. The experiments I set up were solving mazes and attempting to show chemotaxis. Chemotaxis is when a living thing moves towards or away from a chemical. With the solving mazes, the slime molds did not perform very well. Meanwhile, the slime molds seem to have a habit of moving towards toxic chemicals. Overall, the slime molds I collected seem to not be the most intelligent slimes around, but they do show promise in other areas. The slime molds are easy to grow, really easy to work with, and they're a fun way to get into microbiology. They're also large see-through cells, which are perfect for demonstrating how cells work. I think with some work, it'll be possible to get these amazing organisms into research labs and classrooms around the country. And finally, I need to give a big shout out to the Brian Mason Scientific and Technical Trust for funding this research. Thank you.